Hold on, I have no volume. Hello, AP Bio folks. Today we are getting an overview of cell signaling. We talked briefly about this yesterday when we talked about the neurotransmitters that carry signals from one neuron to the next. That type of signaling where a small molecule bonds to a protein and sends a signal between cells is very, very common and very important in living things. So today we're going to look at an overview of this process of cell signaling. This type of signaling helps coordinate different types of activities in multicellular organisms. If you think about something like the onset of puberty in humans, there are lots of different things that happen in a person's body when they reach that developmental stage, right? These types of signals help coordinate that activity so that many different things can happen at the same time or in the correct order. This also happens amongst prokaryotes. Bacteria use a form of cell signaling as well to do things like turn on the ability to produce certain toxins and kind of become virulent, which means causing a disease. And I will leave some resources so you can learn more about that if you are interested. Cell signaling can happen on a few different levels. It can happen when cells are in very close proximity, that's called local signaling, sometimes through direct contact when cells are actually touching each other, like in the immune system. Sometimes when a cell secretes messenger molecules, like those neurotransmitters that we talked about yesterday, that's that synaptic signaling or growth factors. Sometimes if you're injured, cells will send out signals telling the cells nearby to start dividing to heal that wound. It can also happen over a longer distance using things like hormones, your endocrine system that can travel throughout your entire body and carry a system from somewhere way up in your brain down to a further part of your body. So there are lots and lots of different examples of how this type of signaling works. The process of cell signaling is very much like a Rube Goldberg machine. If you're familiar with that mousetrap kind of thing where like you do one thing to start the signal and that makes this thing roll down the hill and bump this, which knocks this over, that falls into that, and it's this whole big long complex thing that leads to one final step. That's how this happens as well. Cell signaling has three basic steps: reception of the signal. Transduction is kind of that whole middle part of the Rube Goldberg machine where this molecule changes shape, which affects this one, which passes the signal to this one, which bonds with this one, which activates the final response. So those three basic stages again. The reception step is when a chemical signal, that signaling molecule is also called a ligand, that word you heard yesterday, bonds to a receptor protein that could be in the membrane or could be inside the cell. Transduction is the middle part where there's a series of steps that converts the signal into a different form. So it's a series of chemical changes that involve several different molecules. This part is also called a signal transduction pathway. And the final step is the cellular response. And it could be a lot of things. It could be telling the cell to produce some enzymes. It could be activating transcription of a particular gene to make more of a protein. It could be rearranging the cytoskeleton. There are lots of different responses that can come from this. So we're going to look at each of these three steps in a little more detail. Reception is the first part. And there are many, many different signaling molecules in your body. Like we said, all kinds of hormones, all kinds of neurotransmitters, and many other molecules can act as those signaling molecules, those ligands. But only cells that have the correctly shaped receptor protein will be able to receive that signal. And that's why your body can send hormones throughout your entire system, but the only parts of your body that respond are the ones that have that correct receptor protein. So these signals are very specific. They bind to that receptor protein, and like any bonding with a protein, anytime you change the chemistry near a protein, it causes that entire protein to change shape. That binding, like we said, is very specific. That's kind of that key and lock analogy again. And it causes a conformational change. That is just another fancy way to say that it changes the shape of that receptor protein, which then transmits that signal into the cell. Some of the receptor proteins are in the cell membrane. So a transmembrane receptor is one of those proteins that's in the membrane of a cell. In this case, the ligands, those signaling molecules, are generally water soluble, which means they're hydrophilic. And so they don't want to pass through the cell membrane, that hydrophobic interior. They don't cross that very easily. And so these ligands bond on the outside of the cell, or they're fairly large and can't fit through the cell. One really common example of this type of transmembrane receptor is called a G protein. This is a class of proteins. There are a whole bunch of them. The ligand, the signaling molecule, bonds on the outside of the membrane here, which causes this protein to change shape and transmits the signal and causes changes to molecules within the cell. I'll link an animation that you can watch that shows how this works, so that you can kind of see how this happens. G proteins are an important group to at least have heard of, because they're used by lots of different types of signaling molecules. And this group of proteins is found in many, many organisms, which shows they probably evolved pretty early on in the evolution of life because they're very common. They have a whole bunch of different functions. They help with different steps of embryonic development. They help with our senses and taking in information that way. And these also are a place that we see a lot of interference in some diseases, things like cholera and pertussis and botulism. Those diseases are caused when something interferes with the functioning of these G proteins. So that's an example of a receptor protein that is found in the cell membrane itself. Another example that we talked about yesterday is a ligand-gated ion channel. And these are the ones that we looked at. This is what happens with a lot of neurotransmitters when they bond to the postsynaptic membrane. The binding of the ligand changes the shape of the receptor in such a way that it opens that protein, there you can see that picture on the bottom, that allows an ion to diffuse. So this is very common in the nervous system, and you find these other places as well. So that's another type of transmembrane receptor. Other receptor proteins are actually inside of the cell. So sometimes that signaling molecule will diffuse through the cell membrane and actually enter the cell and bind with a protein there to transmit a signal. In this case, most of the ligands are hydrophobic so that they're able to diffuse through that membrane. And a lot of steroid hormones, remember steroids are a group of lipids, are able to diffuse right through the membrane and bond with proteins inside the cell. So these receptors are either in the cytoplasm or they might be all the way into the nucleus. And often these types of signaling molecules trigger the transcription of genes. They tell the cells to either start transcribing and translating a particular gene or stop doing that. So they control that process and kind of how your cell uses the information in the DNA. So that's the reception part. Whether the receptor protein's on the membrane or inside the cell, it changes shape and it starts this cascade of molecular interactions in that transduction part in the middle. These are often a multi-step pathway, and you'll see a lot of diagrams that look really confusing, but there are a few common elements that you should always look for. Often these pathways include activating proteins by adding and removing phosphate groups. That's called phosphorylation. Phosphate groups are a really important way that cells transfer energy. Another really common thing is the release of other small molecules or ions that act as what they call second messengers and kind of carry that message to the next molecule. During this pathway, a lot of times you'll see what's called signal amplification. Amplification means getting bigger. Sometimes one signaling molecule can create lots of second messengers and kind of make that response larger within the cell. The reason these pathways are so complex is that at every step, your cell has the ability to kind of control how much of a messenger is made or destroy some of those messengers. So there are lots of places that we can kind of interfere with this pathway and control how these signals are sent. So it allows for a lot more diversity in terms of the types of responses your cells can have. Remember, the molecules themselves are not just traveling through the cell. The signal is being transferred, just like in a Rube Goldberg machine. Each little bit moves a little ways, but the signal continues to move through several different things. So there are lots of molecules, usually proteins, that are involved in this pathway. It's not just one thing traveling through the cell. So that protein phosphorylation, remember I said one of the common things in this multi-step pathway is that idea of phosphorylation. There are a couple of really common enzymes that you'll see. One's called a protein kinase that transfers, it adds a phosphate group onto a protein. That changes the shape of that protein because phosphate groups are negative, and so that changes the whole shape of that protein and activates, it kind of turns on that protein. So protein kinases phosphorylate other proteins to turn them on. Often you'll see this happening several times in a row, and we call that a phosphorylation cascade. So if this happens over and over, that's a phosphorylation cascade. Another enzyme that you'll frequently see is a protein phosphatase. That's like the off switch. It removes that phosphate group and turns that protein back off. So here's an example of a phosphorylation cascade. Here's our ligand signaling molecule. This one's a transmembrane receptor. When this protein changes shape, it activates this first molecule, and this protein kinase then activates, it gets that phosphate, it adds a phosphate group to another one and phosphorylates this one and activates that, which then phosphorylates this protein and activates it. So it's turning on, then turning on the next, then turning on the next. Meanwhile, these PP ovals, those are those protein phosphatases, those are the off switches. And so after this protein has done its job, the protein phosphatase takes that extra phosphate off and inactivates it. So this can be kind of turned on and off and reused. So this is kind of what a phosphorylation cascade looks like. Another thing that's really common within these pathways is what are called second messengers. They're usually really small non-protein molecules or ions. A lot of times calcium ions are used as second messengers, and there are some others that you'll see. They spread really quickly through the cell by diffusion. So like I said, calcium ions, another one is called cyclic AMP. This is a molecule related to ATP. This is adenosine monophosphate. That's another really common second messenger. So anytime you see these types of molecules in a pathway, they are the second messengers, and these really help with that signal amplification idea. One signaling molecule, so as you can see in this example, the ligand is up here at the top. That signaling molecule changes this molecule and triggers this to release a whole bunch of cyclic AMP. And it only shows one, but every single one of these molecules then is going to go out and activate this pKa molecule that activates a bunch of protein kinases. And so you can see the signal is just getting bigger and bigger and bigger and spreading through the cell. And eventually those signals reach their destination. They go through all those transduction steps and they trigger a response within the cell. If that response is in the nucleus, then it usually has to do with turning genes on and off, which regulates the process of protein synthesis. So it determines which proteins are being built in a cell and how much of those proteins are being built. If it's a response in the cytoplasm, then it usually regulates the activity of proteins. So it might turn off proteins and prevent them from working or inhibit the functioning of an enzyme. Instead of the actual building, it kind of regulates what they're doing. So you are going to be working on a project over the next couple days, looking at one specific example of a cell signaling pathway. During the live session today, I'm going to put you into groups with a partner and you will be choosing a topic. And so if you're not able to attend the live session after class this afternoon, I will contact you or one of your classmates will contact you to let you know the details of how this project's going to work. But I just want to show you what this project will look like. There will be a slide presentation that's attached in the homework section in Classroom that looks like this. Everyone in the class is going to be working in the same presentation, but you are only responsible for editing one slide. Your task here is going to be to research one signaling pathway that happens in living things and complete one slide about your pathway, which you will present to the class either Friday or Monday during the live session. Your group will pick from one of these signaling molecules, and these are a whole variety. Some of these are neurotransmitters, some of them are hormones, some of them are plant hormones, and some of them have to do with the immune system. So there's a whole variety of examples that we're going to look at here. Once you have selected your topic, you will find a slide that looks like this, and you'll find the one that has your signaling molecule in there. And then you're going to be filling in this information. You can delete what's in parentheses down here, and you're going to write about the source of your signal. So where is that signaling molecule produced? What type of molecule is it? Those types of things. So the stuff in parentheses just gives you ideas of what I'm looking for in that box. The video link part of this is optional. This project was originally designed as a little bigger thing that included like a stop motion video that you had to make. We're not doing that this year as a requirement, but if you find a good animation or a short video that illustrates how your pathway works, you're welcome to link a video in here, or if you even just want to make your own video kind of talking through how it works, you're welcome to do that and link that in here to show us as part of your presentation. Keep track of your works cited. This doesn't need to be super highly formatted, but at least a list of websites and resources that you used so I know where you got your information from. Tomorrow is Wednesday. You will have all day to work on this project. That's your main task tomorrow. You do also have mastering biology homework due tomorrow night if that's not done yet. These projects need to be done before class on Friday because that's when we will be starting the presentations. Contact me if you have any questions, and I hope you have a great rest of your day.